For billions of years, an invisible, relentless war has been raging on a microscopic scale. Bacteria have been under constant assault from invading viruses known as bacteriophages. To survive this endless onslaught, these bacteria developed a highly classified defense system. They began capturing small fragments of the invading virus's DNA, storing them as a genetic memory bank to identify and destroy those exact attackers if they ever returned. We call this ancient immune system CRISPR. CRISPR stands for clustered regularly interspaced short palindromic repeats, which is the structural pattern in bacterial DNA that houses these stolen viral mugshots. The real twist to this story happened in 2012. Human scientists pulled off the ultimate biological heist, taking this bacterial defense mechanism and reverse engineering it for our own use. By commandeering this ancient weapon, researchers secured a precision search and replace tool for life itself. This physical model shows the twisting, ladder-like structure of the double helix, the framework that holds our genetic code. Built upon this physical structure is the genome. The genome is the complete, comprehensive genetic instruction manual required to build and maintain a living organism. In humans, this manual is staggeringly large. It consists of a 3 billion-letter sequence that dictates everything from your eye color to your risk of developing certain diseases. Gaining the mechanical ability to pry open this specific database and edit the text inside grants us direct control over biological destiny. To understand how this break-in works, we have to look at the two distinct pieces of machinery that make up the CRISPR-Cas9 system. The first component is guide RNA. This programmable molecular bloodhound carries a custom genetic snippet to find an exact DNA match. The second piece is the Cas9 enzyme, microscopic scissors that lock on to the guide RNA, armed and ready. Once deployed inside a cell, this combined complex slides along the DNA strands, reading the rungs like a microscopic barcode scanner checking for a match. Together, these two parts form a highly accurate biological search engine, capable of hunting down a single genetic typo hidden among 3 billion normal letters. Finding its exact genetic match, the complex stops. It physically pries the two target DNA strands apart and docks securely into the gap. This triggers the Cas9 scissors. The enzyme cleanly severs both strands of the target DNA, breaking the genetic code at a precise location. A severed DNA strand is catastrophic for a cell. In response, it immediately hits the panic button and activates its natural, frantic DNA repair mechanisms to fix the break. Here is where scientists execute the final trick of the heist. By flooding the site of the break with a custom, corrected piece of DNA, they trick the panicked cell. It immediately grabs this new genetic material and blindly uses it as a patch to bridge the gap. Scan, sever, and patch. This exact 3-step sequence is the physical reality of how we rewrite the traits of a living organism. For decades, science was limited to simply observing and mapping the genetic code. We have now crossed the threshold from passively reading DNA to actively writing it. The medical implications of this are immense. We can target the exact genetic mutations responsible for debilitating conditions and correct the code directly at the source. As we can see on this requisition form, technologies like CRISPR-Cas9 are already moving through clinical trials to treat single-gene disorders like sickle cell disease. Conditions that were long considered permanent, inescapable errors hard-coded into our biology are now entirely treatable. But looking at these early stages of embryo cell division, we draw a strict ethical boundary. Editing somatic cells, like the blood cells in a single adult patient, is drastically different from editing the cells of an early human embryo. Editing an embryo is known as germline editing. Germline editing means making genetic alterations to egg or sperm cells that do not just affect one individual, but are permanently passed down to all future generations. Taking control of the permanent course of human evolution carries extreme risks. It is a level of power that demands profound respect, caution, and rigorous global oversight. To camera, I'm the Autodidact. Thanks for learning with me in the lab.